Thanks everyone in the GU Oncology Now community. We're so happy that you've joined us. I'm thrilled to be here with two wonderful colleagues and friends of mine. First and senior author on an exciting paper recently published in the JCI publication, and just really detailing more a better understanding of HIF biology. And I think there's a lot of excitement right now around targeting HIF biology with a novel treatment option for patients with kidney cancer. So I'm excited to welcome Zach, first author of this paper, and Jeff, senior author. So Zach, maybe have you start off by introducing yourself, and we'll go from there.
Sure. Thanks, Katie. So I'm Zach Bacigalupa. I began in 2018 in Kim Rathmell's lab, but was also co-mentored by Jeff as a postdoc, and have for the past almost 6 years now been really working on this project as my primary project, and we are delighted to be able to share it with everybody now.
That's great. And Jeff, I know we're all part of this same team here, but would love for you to introduce yourself. And
Sure. Yeah, Jeff Rathmell, professor and direct the Center for Immunobiology. I've been working with Zach here since he came and joined the group a few years ago. We've been interested in, on one hand, immune function and immune metabolism, but that really stems from a broader interest in cancer metabolism, and kidney cancer has been an area of focus for us. So this paper sort of launches in that direction.
Great. Zach, maybe start us off by kind of laying the landscape. Why did you become interested in essentially investigating ACSS2, and why is it important?
Yeah, so it was actually really great timing. So when I was in graduate school, I'd studied acetate metabolism in ACSS2 and glioblastoma. And just being a grad student, I was reading some papers that had come out previously from Joseph Garcia's lab, who had kind of laid some groundwork that was describing and just fundamental biological systems, not necessarily cancer, but that there was a link between acetate metabolism, ACSS2, and HIF-2. And it was very specific about HIF-2, not necessarily HIF-1. And at the time, I hadn't really thought much of it because it didn't apply to my system in that setting. But when I joined down here, I learned very quickly that HIF-2 is centric to driving clear cell renal cell carcinoma. And a lot of light bulbs went off my head. And I said, wait, I'm going to be studying acetate metabolism again. So it just seemed like a very clean story, which there ended up being a lot more mechanistic detail that come out of it. But it just seemed to be in line with kind of what I was used to studying. I had some tools and methods to go about getting at this project pretty quickly, and addressing a really important need in the kidney cancer community.
That's great. And maybe for our audience here who may not be familiar with all the details, and some clinicians and some medical colleagues here, what were your main findings in this paper?
Well, I think first and foremost was that we start off in figure 1 where we did some single cell. We analyzed some single cell RNA-seq data sets because the bulk RNA-seq data sets suggested that ACS2 was actually had low expression. But kidney cancer is so heterogeneous. There's so many different cell types that infiltrate, particularly immune cells. And when we looked at the single cell data set, we actually saw that ACS2 expression was very low in the immune cells, but quite high in the cancer cells. So I thought that was really important, kind of on a broader scale, that a lot of different cancer biologists can take into perspective that we do need to dig a little deeper. These bulk data sets are really useful, but sometimes you do need to go a little bit deeper to find something that might be really important to the question that you're seeking to answer. So I thought that was a really important thing. The other important element that I thought came out of this was that ACSS2, it's been described as a really good therapeutic target in breast cancer, brain cancer, a couple of others as well. But no one had really looked at it in the context of kidney cancer. And just given the link to HIF-2 biology that the Garcia lab had explored previously, but had not looked at in kidney cancer, drawing that link was so huge, and seeing that it did impact, if we inhibited ACSS2 genetically or pharmacologically, we could block tumor growth pretty effectively. And really, when we looked at how we were regulating HIF-2, it was through two different mechanisms. It was both at the transcriptional level through what we believe is some epigenetic mechanisms. ACS2 acetate metabolism is going to touch on histone acetylation. So we were able to see that with ATAC sequencing. But we also see that we were able to alter HIF-2 protein stability. So you're kind of having two mechanisms at the same time to regulate HIF-2, which is different than anything else that's been offered, at the clinical level.